When you try to picture the ultimate secret to human life, I mean, the master blueprint that builds everything from your brain cells to your bones, you'd expect it to look, well, impressive.
Right, like some kind of overwhelmingly complex biological supercomputer or something.
Exactly. But if you were a scientist in the early 20th century, looking right at the very molecule that does all this, you probably would have walked right past it.
Yeah, you really would have.
For decades, the absolute brightest minds in biology looked at DNA and collectively decided it was just, like, too boring to be the secret of life.
They saw this monotonous, repetitive chemical and thought, there's no way this simple thing holds the instructions for a human being. It's just impossible.
Right. So welcome to today's deep dive. We have a massive stack of source material today.
Huge stack.
Yeah, it's an incredibly comprehensive educational text on the molecular and historical foundations of genetics. We're tearing it down to the studs today.
We really are.
This is custom tailored for anyone prepping for their TEAS and HESI nursing entrance exams, specifically looking at how traits are passed on. But honestly, if you have a body, this is going to be fascinating for you.
Absolutely. We're going to demystify how a simple 4-letter chemical alphabet builds a human, where things go wrong, and why understanding this is the absolute bedrock of modern healthcare.
Okay, let's unpack this idea that DNA was considered boring. What exactly were these early scientists looking at that made them so unimpressed?
Well, they were looking at its chemical structure. DNA, deoxyribonucleic acid, is just a polymer made of repeating units called nucleotides.
Okay.
And each nucleotide is just a phosphate group, a sugar, and one of 4 bases: adenine, thymine, guanine, and cytosine. So A, T, G, and C.
So they were basically looking at a 4-letter alphabet.
Right.
And I guess, if you're trying to write the insanely complex instruction manual for a human being, and you look over and see that proteins are made of 20 different amino acids, you'd naturally assume the 20-letter alphabet is running the show.
Exactly. That was the protein hypothesis, and it kind of ruled science for a long time. People thought DNA was just structural scaffolding.
Just keeping things held up.
Yeah, exactly. It took a bizarre historical detective story to prove otherwise, starting with Gregor Mendel.
Ah, the pea plant guy.
Right. He was breeding pea plants in a monastery in the 1860s, meticulously tracking traits like pod color and seed shape.
Right, the classic green and yellow peas.
Exactly. And he realized heredity worked in these predictable mathematical ratios, and he called these traits invisible factors. So he publishes his work.
And let me guess, everyone loved it.
Not at all. The scientific community essentially ignored it for over 3 decades.
Man, they just completely ghosted the father of genetics.
Pretty much. Meanwhile, we get our first physical glimpse of DNA, and the origin story is wonderfully gross.
Oh boy. How gross are we talking?
Well, a Swiss physician named Friedrich Miescher wants to study the chemistry of cells in 1869. So he goes to a local surgical clinic, takes their discarded, pus-soaked bandages.
Oh, wow. Oh,
Yeah, and scrapes off the white blood cells from the pus.
that is deeply unpleasant.
It is. But from the nuclei of those pus cells, he isolates this mysterious phosphorus-rich substance. He calls it nuclein.
Nuclein. But he didn't know what it did.
No clue. He had no idea it had anything to do with Mendel's invisible factors. To him, it's just a weird chemical. It takes decades and some incredibly strange experiments to finally connect the dots.
Like what? What finally proved it?
Well, take Frederick Griffith's work in 1928 with bacteria. He found that if you take harmless bacteria and mix them with the remains of heat-killed lethal bacteria...
Wait, dead bacteria.
Right, dead lethal ones. The harmless ones suddenly transform into deadly ones, and they pass that deadly trait onto their offspring. Something physical was being transferred.
The transforming principle, right.
Yep.
But they still didn't know if that something was a protein or DNA.
Exactly. Oswald Avery and his team in 1944 eventually cracked that part. They took that lethal mixture and used specific enzymes to destroy one type of molecule at a time.
Oh, that's smart. Process of elimination.
Right. Destroy the proteins. The bacteria still transform. Destroy the RNA. They still transform. But when they destroy the DNA...
The transformation stops dead in its tracks.
Exactly. DNA was the culprit. But scientists still didn't believe it because of that deep-seated bias toward proteins.
They just couldn't let it go.
Nope. So the final nail in the coffin was the Hershey-Chase blender experiment in 1952. They used radioactive tracers to tag viruses.
How does that work?
Well, they tagged the virus's outer protein coat with one radioactive marker and its inner DNA with a different one. When the virus infected a bacterium, Only the radioactive DNA went inside to hijack the cell.
And the protein coat just stayed outside.
Right. And to prove it, they actually used a literal kitchen blender to knock the empty protein coats off the bacteria.
A kitchen blender. That's amazing. The ultimate proof it was DNA all along.
Yep. And then we get the legendary race to find its structure, ending with James Watson and Francis Crick publishing the famous double helix model in 1953.
Though I want to point out, our sources make it very clear they relied heavily on Erwin Chargaff's chemical pairing rules and Rosalind Franklin's X-ray diffraction image.
Ah, yes, photo 51.
Right, the photo which literally captured the shadow of the helix. Her tragic death before she could be recognized with a Nobel is, still a heavily debated omission in scientific history.
Absolutely. But what her photo and Watson and Crick's model revealed, it changed biology forever.
Okay, so they proved it looks like a twisted ladder. But a shape alone doesn't explain how a parent passes traits down to a child. How does this massive ladder actually copy itself without making millions of mistakes?
It all comes down to what our text calls the base pairing rule. This is the entire mechanism of heredity in one sentence. Adenine only bonds with thymine, and guanine only bonds with cytosine.
So A to T, G to C, always.
Always. Because of this strict rule, if you have one half of the DNA ladder, you can perfectly predict the other half.
It's kind of like a zipper. When a cell needs to divide, an enzyme comes along and just unzips the double helix right down the middle.
Right, exactly. Now you have two single strands just flapping in the breeze. But because A only wants T and G only wants C, floating nucleotides in the cell naturally pair up with those exposed single strands.
So you end up with two complete double helices.
Right, and each is made of one old strand and one brand new strand. The scientific term for this is semi-conservative replication, proved by Michelson and Stahl in '58.
Semi-conservative.
Okay, yeah, and this zipper mechanism is exactly why a parent's cells can produce a perfect copy of their genetic library to hand down.
Let's ground this in the body for the listener. There are two main ways this copying is used. When you scrape your knee, your skin cells use a process called mitosis.
Right.
They copy their DNA and divide into two identical cells with the standard 46 chromosomes to heal the cut. But passing traits to a child is totally different.
Completely different. That requires meiosis, which happens in the testes and ovaries. It deliberately halves the genetic material.
So instead of 46 chromosomes, you get, what, 23?
Exactly. It packages just 23 single chromosomes into a sperm or an egg cell. That way, when fertilization happens, 23 from the mother and 23 from the father combine.
To restore the full set of 46 in the offspring. That makes perfect sense. Meiosis is basically the physical bridge between generations.
It is.
Okay, so we have this vast library of code, these 46 chromosomes. We know how it copies itself. But I have to push back here on the analogy.
Okay, go for it.
A library doesn't actually build a city. A book just sits on a shelf. How does this static code of A's, T's, G's, and C's actually go out and do things in a human body?
That brings us to the central dogma of molecular biology, coined by Francis Crick. It's the directional flow of information, DNA to RNA to protein.
DNA to RNA to protein. Okay, explain that.
Think of your DNA as the master reference book locked inside a high-security vault, which is the nucleus of the cell.
Right.
It is way too valuable to leave the vault. So when the cell needs to build something, say a digestive enzyme or a muscle fiber, it does a process called transcription.
Transcription, like making a copy.
Exactly. It unzips just that one specific gene and makes a disposable blueprint out of a cousin molecule called messenger RNA.
Okay, mRNA. So this mRNA blueprint is smuggled out of the vault and heads out to the construction site, the cytoplasm. Does it just magically assemble a protein on its own, or is there a builder?
Oh, there is definitely a builder. That's the second step translation. The mRNA blueprint is fed through a cellular 3D printer called a ribosome.
A ribosome.
Right. And the ribosome reads the mRNA letters, but it reads them in groups of 3, called codons. Every single 3-letter codon corresponds to one specific amino acid.
So it's translating the nucleic acid language into the amino acid language.
You got it. As the ribosome reads the blueprint down the line, codon by codon, it grabs the matching amino acids and links them together into a long chain.
And that chain is the protein.
Once the chain is finished, it folds up into a complex 3D shape. That is a protein. And proteins are what actually do the physical work in your body.
Okay, so from a 4-letter code, read 3 letters at a time, you build a 20-letter alphabet of proteins. But I mean, the system is copying billions of letters.
Yeah.
What happens when this biological 3D printer gets a typo in the blueprint?
When the copier glitches, you get a mutation. Our source material uses 2 very distinct genetic diseases to illustrate how different kinds of mutations ripple through a person's life.
Let's get into those.
Let's look at sickle cell disease first. This is a recessive disorder, meaning you have to inherit the mutated gene from both parents to have the disease. And the mutation itself is terrifyingly small.
How small?
It's a single nucleotide substitution. One single letter out of billions is swapped in the gene that builds hemoglobin.
Wow. Which changes one three-letter codon. So the ribosome grabs the wrong amino acid swapping in valine instead of glutamic acid at the exact sixth position of the protein chain.
Exactly.
But how does one wrong amino acid cause such a devastating disease?
It changes the entire 3D shape of the protein. Under low oxygen conditions, those slightly misfolded hemoglobin proteins suddenly lock together.
Like they stink to each other.
Yeah, they form these rigid, spiky rods inside the red blood cell.
Yeah.
It distorts the cell from a nice, smooth donut shape into a sharp sickle shape.
Ouch.
Right. And those jagged cells get stuck in tiny blood vessels, causing excruciating pain, organ damage, and chronic anemia.
That sounds awful.
Yeah.
But I have a question. If sickle cell is so devastating to human health, why hasn't natural selection just wiped that ill out of the human population entirely?
Well, it reveals one of the most counterintuitive truths in genetics. We mentioned you need 2 copies of the mutated gene to have sickle cell disease, right?
Right.
But if you only have 1 copy, you are a carrier. You don't have the disease, you are generally healthy, but your red blood cells are just a tiny bit different.
Okay.
And having just 1 copy of the sickle cell allele provides a strong resistance to severe malaria infection. The malaria parasite actually struggles to survive in those slightly altered red blood cells.
Oh, wow. I didn't know that.
Yeah, so in regions where malaria is historically deadly, like parts of Africa, the Mediterranean and South Asia being a carrier meant you were more likely to survive childhood and pass your genes on.
So a mutation that is catastrophic in a double dose is actually highly beneficial in a single dose.
Exactly.
The environment completely dictates whether a mutation is a curse or a shield. What about the other example from the text? Huntington's disease.
What's fascinating here is how this compares to sickle cell. Huntington's is dominant.
Meaning you only need 1 copy.
Right. Inherit just 1 copy of the mutated gene from either parent and you will develop the disease. And the mutation isn't a single letter swap.
What is it then?
The text calls it a stutter, a trinucleotide repeat expansion. In the Huntington gene, there is a specific 3-letter sequence, CAG, that normally repeats a few times.
Okay.
But in Huntington's, the cellular copying machinery literally stutters. It copies CAG over and over and over again.
Expanding the sequence way beyond its normal length. So instead of 1 wrong building block, the 3D printer is forced to just keep adding the same block endlessly.
Exactly.
Why is that bad?
Because it creates a toxic, abnormally long protein. These mutant proteins are unstable. They break down and clump together inside the neurons of the brain.
That doesn't sound good.
It's not. Over time, these physical clumps literally destroy the brain cells, leading to the severe neurological symptoms we see in Huntington's patients.
It's just so mechanical.
Right.
A swap versus a stutter. We tend to think of our DNA as this fixed, unyielding script. We think DNA is destiny. But our source text tackles this head on.
It does.
Here's where it gets really interesting.
Yeah.
We need to bust some major misconceptions.
The first big one is the idea that DNA and genes are the exact same thing. They aren't.
They aren't.
No. DNA is the physical molecule. A gene is a functional segment of that molecule that actually codes for a trait. But over 98% of human DNA does not code for proteins at all.
Wait, 98%? But evolution is usually brutally efficient. Why would the body spend massive amounts of energy copying billions of letters of junk DNA for millions of years? Didn't that seem off to scientists?
It absolutely should have. Geneticists now realize calling it junk was wildly misleading, that 98% contains crucial regulatory sequences.
That's what they do.
They act like volume knobs, basically, turning nearby genes up or down or telling the cell exactly when to build a certain protein.
Oh, that makes a lot more sense. Another huge misconception comes from Mendel's peas, where 1 gene equals 1 trait. Green pod or yellow pod.
Right, but human traits are rarely that simple. Height, skin tone, intelligence, susceptibility to most diseases. These are polygenic.
Meaning many genes.
Exactly. They are faked by dozens, sometimes hundreds of genes, all interacting with each other and interacting with your environment, producing a massive continuous spectrum of outcomes. It's not just an on-off switch.
And just because a disease is genetic doesn't mean it was inherited from your parents, right?
Correct. Many mutations are what we call de novo. They happen spontaneously in a specific egg or sperm cell or very early in embryonic development.
So neither parent has the mutation, but the child does.
Yep. Genetic just means it involves the DNA. It doesn't automatically mean it was passed down from the previous generation.
Right. But the most mind-bending concept in the text for me is epigenetics. The idea that your environment, your diet, your stress levels, your exposure to toxins can actually leave chemical marks on your DNA.
We really need to be clear on how this works. These epigenetic marks do not change the sequence of the letters. The A, T, G, and C stay exactly the same.
So what changes?
They are physical chemical tags that attach the DNA molecule and basically block the 3D printer from reading it. They can turn a gene completely off or crank it into overdrive. The best proof of this is identical twins.
Right, because at birth, identical twins share virtually 100% of their DNA sequence.
Exactly. But as they grow, they experience different environments, different stresses, different diets. Their epigenetic marks diverge.
So by the time they're in their 70s, their gene expression, which genes are actually turned on and off, can be vastly different.
Completely different. Genetically identical does not mean biologically identical forever. And some of those epigenetic marks can actually be passed down to the next generation.
That radically expands our definition of heredity. It's not just the hardware you pass on, it's the software settings too.
It is a profound shift in how we understand biology. But, whenever we talk about understanding and controlling human heredity, this raises an important question.
Right, and the source material does not shy away from the dark history that accompanies this science, specifically eugenics.
Yeah, eugenics was a movement in the early 20th century that took very crude, often entirely false ideas about heredity and essentially weaponized them.
The goal was to improve the human population by preventing people deemed unfit from reproducing, right?
Yep.
Usually targeting people based on poverty, disability, race, or ethnicity.
Exactly. And it wasn't just fringe science, it was state-sponsored.
Yeah, the text specifically highlights how the U.S. Supreme Court, in the case Buck v. Bell, actually upheld state laws that allowed for the compulsory sterilization of citizens. Tens of thousands of Americans were forcibly sterilized.
It's horrific. And as the text notes, for anyone entering the health sciences, this is essential context. It is the ultimate historical warning of what happens when a real biological concept is twisted into pseudoscience to justify systemic harm.
Well said. We're just reporting what the sources outline here, but it's a vital part of the history.
Absolutely. And while the eugenics movement of the 1920s is behind us, the ethical debates about controlling heredity are more intense than ever because our technology has caught up with our imaginations.
The text highlights two major modern milestones here, right?
Yes. In 1996, Dolly the sheep was born in Scotland. She was the first mammal ever cloned from an adult cell.
Proving that the DNA inside a fully mature adult cell still possessed the capacity to reboot and build an entire new organism from scratch.
Exactly. Then we get to gene editing, CRISPR. In 2018, Chinese scientist He Jiankui shocked the world by announcing he had used CRISPR to alter the DNA of human embryos.
Resulting in the birth of twin girls, right?
Right. CRISPR works like programmable molecular scissors. You can guide it to a very specific sequence of DNA, cut it out, and paste in a new sequence.
But doing this in human embryos crosses a massive line.
A huge line. Because those edits become part of the germline, meaning those little girls will pass those artificial edits down to their own children.
And the scientific community globally condemned the experiment as a massive breach of safety and ethics. And He Jiankui was imprisoned. It perfectly illustrates the modern chasm between the fact that we can edit human heredity and the profound debate over whether we should.
Which brings us to what this all means for you listening. Especially if you are heading into nursing or healthcare, this isn't just historical trivia to memorize for a test.
No, this is the reality of patient care today.
You will be dealing with genetic counseling, helping prospective parents understand the recurrence risks for conditions like cystic fibrosis. You'll be looking at newborn screening programs, which test infants for dozens of heritable conditions at birth.
Because catching a metabolic error early can literally save a child's brain from damage.
Exactly.
So what does this all mean? The text also brings up pharmacogenomics. The idea that two patients with the exact same diagnosis might need totally different drugs or different doses simply based on how their specific DNA metabolizes the medication.
Personalized medicine is here. And to bring it completely full circle, remember that single-letter typo that causes sickle cell disease.
The one we talked about earlier.
Right. In December of 2023, the FDA approved Casgevy. It is the first CRISPR-based gene therapy for sickle cell disease.
Wow.
Yeah, we are now using those molecular scissors to go into a living patient's stem cells and physically fix the expression of hemoglobin.
The journey from Gregor Mendel, counting yellow and green peas in a monastery garden, to doctors curing a genetic disease by editing the code of life inside a patient. I mean, it's just breathtaking.
It really is.
We started this deep dive talking about how scientists thought the DNA molecule was just a boring static instruction manual, a book sitting in a vault.
Yeah.
But I want to leave you with a final thought to mull over.
What's that?
If our lifestyle and environment can create epigenetic marks that we pass down to our kids, and if we are now actively deploying tools like CRISPR to physically edit our own genetic code in real time, are we no longer just the passive inheritors of our DNA? Are we becoming the active authors of it? And if we are writing the manual as we go, what does the word heredity even mean for the next generation?